A new time of personalized medicine has come, with certain medical therapies tailored to individual patients and their own genes. This practice is being used in cancer treatments. To create the individualized treatments, they sequence a patient's entire genome, which should lead to the crucial information about the person's cancer and possibly lead doctors to a specific therapy. Sequencing the whole genome identifies changes to patient's DNA that would not have shown up on standardized genetic tests, researchers said. Personalized sequencing impacts cancer in several ways, the first being DNA sequencing. One of the first to apply this study was to analyze the DNA of an acute myeloid leukemia patient, in which both tumor and normal cells were sequenced. Mutations were found in the tumor's DNA, but none in the normal DNA. Two of the mutations found were linked to acute myeloid leukemia and the others were novel. This specific study put forth a tool to discover the novel mutations and the potential targeted treatments. One of the biggest outcomes of the discoveries found with sequencing was that most cancers are very different from one another, yet common mutated pathways can be observed. Personalized tumor DNA sequencing can directly impact treatment by identifying mutations that can lead to treatments. In some cases, the information found using DNA sequencing can identify a pathway to pre-existing treatments and possibly even new potential treatments that are yet to be discovered. Personalized sequencing will likely lead to guiding ongoing surveillance of recurrence in certain diagnoses. Challenges of sequenced interpretation. Despite these clear successes, many challenges can make these successes less frequent. It may be difficult to identify the variant in certain cases, filtering the variants independent of the phenotype. If a person's phenotype is too broad or non-specific, then the identification of their genes is complicated to identify. DNA sequencing can cause significant damage to the DNA being tested. Although this seems like the perfect discovery needed to help cure cancer, scientists do not know what to do with most of the information they get from a whole genome sequence, most of it being uninterpretable. In conclusion, even with many obstacles, personalized sequencing has already begun to demonstrate its applicability to the practice of medicine. With it, we have begun to better understand rare and long-studied diseases such as cancer. As implementation of personalized sequencing becomes more achievable, the accuracy is significantly improved.